right, alpha helix. This is one of the commonest hydrogen bonded secondary structures that you will see in proteins. So common that about 30% of all the amino acids in the world in solved proteins are found in alpha helices. We always say it's an alpha helix to differentiate it from two other kinds of helix. Worry about those later. Those are going to be the 310 helix and the pi helix. But just focus on the alpha helix because that is the common one. Very common. They're almost always right-handed, which is to say that if they start here, they're going to go like you're right-handed in that direction as they twist. Yeah, there you go. Linus Pauling predicted that they would exist, and sure enough, they do. Not everything he predicted was right, but he's pretty good at his job. So, all right. Alpha helix is going to have a phi bond of around -60 and a psi bond of around -50 degrees, and so they're going to be in that particular quadrant. So the minus quadrant is down here at the bottom of the Ramachandran plot, and so there they are, right-handed alpha helix in that sort of actually fairly narrow range. Alpha helices can be big. 40 amino acids, not unheard of. There are probably alpha helices that are longer than that. I just haven't heard of them. But people find new things all the time. There's probably one somewhere that's 41. This is a YouTube video that I've updated that is a person explaining the hydrogen bonding in an alpha helix. But essentially, look at that. The reds are oxygens, the blues are nitrogens, and so each time you have a red reaching out over a distance of about 3 angstroms and making contact with that nitrogen, but indirectly. The red oxygen is trying to steal the hydrogen that is covalently bonded to the nitrogen. That's what's actually going on there. So yeah, that's it. That was alpha helix. This is a beta strand. And remember, in nature, you don't ever find one beta strand. There are always more than one beta strand, and they form a beta sheet. Also predicted by Pauling, and once again, he did a good job. Yeah. They are essentially when the polypeptide chain is as stretched out as you can get. And by stretched out, just look at that. So it's not coiled, it doesn't bend, it's like you pulled a little curly phone cord and you pulled it straight. Or you pulled a slinky until it's just a wire almost. So they tend to have a phi bond around -139, a psi bond around 135, so they're going to end up in this top corner up here, which also bleeds over into the bottom of this. That's the generously allowed part of the beta sheet domain up here of the Ramachandran plot. Left-handed helices do exist, but they are rare, and so for now, don't worry about them, but they have their own deal. And that's a YouTube video showing the hydrogen bonding that stabilizes these. Basically, what it's going to be is you're going to have this guy, this oxygen, reaching out, hydrogen bonding to the next strand, trying to basically steal that hydrogen once again from the nitrogen. It's just like Tom and Jerry, never ends. Oxygen's got to try and steal a hydrogen from a nitrogen. That's what's going on in proteins all over the world, every day, all the time, right now. Yeah. Okay, here's your beta sheet, and there's your carbon with the double bonded oxygen. Reaching out, the reds are the hydrogen bonds, and it's mostly trying to steal the hydrogen from there. And you see, not to be outdone, this carbon is going to try and steal the nitrogen from this part of the chain. And so the chains can be either parallel or anti-parallel. Anti-parallel meaning going in opposite directions. Parallel meaning going in the same direction. And so parallel strand has got to like find some way to curl around the back, and then it'll have another one, and it can pair up. And and these would be anti-parallel because you got one going this way, one going that way. So yeah, that's it. The hydrogen bonds tend to be like at a kind of an angle in the parallel ones, and more common are the anti-parallel, if I remember correctly, in protein structure, because you can just have a little turn here at the end, boop, and then it comes back, and then it's going anti-parallel because it's you don't have to wrap around and then make another parallel strand. All right, alpha helix, here it is again. There are certain amino acids that are good at getting out of the way and are more common in alpha helix. And so we can remember these as M-A-L-E-K, like Malik, yeah. It's related to an old, it's like a Babylonian word that comes through in Hebrew and various languages that you would find in the Middle East. It means like king or queen. There are words like that are built on that. So think of King Malik with M methionine, A alanine, L leucine, E glutamic acid, yeah, you remembered, and K lysine. Those are pretty common, or more common in helices than out. The ones that you tend to not find in a helix are the pigs, proline, tyrosine, glycine, and serine. And so, yeah, I can't imagine a helical pig. Can you? No way. Yeah, so there you go. Prolines are very often found at the start of a helix because the proline makes a little right-handed bend in the protein that is being synthesized. And it's right-handed in the sense that we're assuming that this is the N-terminus, amino terminus, and the C-terminus is over here, and proline, yeah, puts that little bend in there. You will often find the two acidic amino acids at the end of a helix. They are helix breakers, but they're like helix cappers as well. So there you go. And there are also amino acids that are more common in beta strands. Now, the thing to remember is you will occasionally find any one of these amino acids in any structure, pretty much. There is enough variability in biology that everything happens occasionally. But if we're talking about things that happen a lot, yeah. V, W, I, F, Y, and sometimes T. So TV Wi-Fi. Imagine your television at home has Wi-Fi. W-I-F-Y in this case. Yeah, we're spelling it wrong. And there you go. You've remembered it. Prolines, once again, are going to introduce a very strong sort of right-handed bend. So prolines are not going to be allowed because you can't be bending sharply to the right and extended sort of as far as possible. And so what you find are prolines at the end of sheets, and they will actually make a little turn quite often. You'll have prolines in the orange circled positions. They'll be common at the edge of a strand, which ends up meaning they line up at the edge of a sheet once you have a sheet, because a sheet is just two or more strands that are hydrogen bonded together. Yeah. And you can have lots of strands. I've seen 8 or 10, but I think there are much longer ones than that, where they're all hydrogen bonded in a row, often sort of twisting a little bit. Yeah, kind of interesting. Okay, other types of secondary structure. You can have a 3-10 helix. So normally an alpha helix is going to have 7 amino acids in two turns. Yeah. 3-10 helix has 3 amino acids in each turn. So there's 10 atoms between the hydrogen bond donor and acceptor, basically, is how it works. These are generally rare. They are not particularly favorable, but when the environment around them is just right, this will happen. They tend to be very short, but you can find these, and they are, when a protein has one, it will always have a little 3-10 helix. Other helix you find, and this is another very unfavorable, kind of rare one, but it does occur, yeah, enough that it has a name, and it's called a pi helix. And so, yeah, this is a very long hydrogen bond that makes a very open, wide helix, whereas it was a very tight, narrow helix in the 310 compared to the normal alpha helix. And then this is like a big expanded helix, just like that. Again, fairly rare, but pi helix does exist. Yeah. Okay, so if you dare describe these protein structures, what do you see? Yeah? It's like Dora the Explorer here, right? Yeah. Ah, very good. Very good. Yeah, so, yeah, I see 4 nice orange. So what would we call those? So Beta strands, yeah, but what kind of beta strands are we talking about? Look, the nice thing with beta strands is they'll usually draw them as a flat sort of ribbon, but with a little arrowhead that shows you which way it's pointed. So this one I don't see an arrowhead here, which means the arrowhead's up here. And this one I see the arrowhead and it's right there. So one's going up, one's going down. Those are anti-parallel. That is an anti-parallel, two anti-parallel beta strands. And look at these two. That one's going down and that one's also going down. Those are parallel beta strands. And then over here, you have a little anti-parallel. So you could think of this as two little anti-parallel beta sheets, or one big beta sheet that has both parallel and anti-parallel elements in it. And who's that hanging out in the back, the big coily one? Yeah, take a guess. Yeah. It's an alpha helix, right? Yeah, of course it is. So there's our alpha helix. Look at the blue regions highlighted at the end of the protein. So these are areas where there's a hard right turn, basically. And so these are called turns. The turns involved here can actually be called beta turns because they're within a beta sheet. And this is also going to be a turn going into this beta sheet. And so you're frequently going to have either a glycine or a proline or one or two of each of those that will make a nice rigid turn. Then you got the white stuff, this sort of floppy white stuff. That's going to be random coil. Because everything that's not alpha helix, not beta strand, not weird helix like 310 helix or left-handed helix or pi helix, we're going to call it, and it's not a turn, yeah, we're going to call those random coil. All right, now let's look at the other one over on this side. What do you see? I see 1, 2, 3, short little 4, and probably 5 alpha helices. I'm counting this a little bit up here as separate because an alpha helix is generally a straight rod, maybe with a little bend to it, and it never bends like an L, so that would be 2 alpha helices. They're just kind of on top of each other. And what's all that stuff in between them? Why, that's random coil, my friends. Yeah, of course it is. How can it be anything else? Those random coil bits, or loop regions, are areas where you get a lot of genetic variability because the things that are hydrogen bonded are actually holding the protein together in a particular shape. The loops are just there so that you can turn and make that next, anti-parallel beta strand, or so you can turn and get that next alpha helix going where it can interact with the rest of the molecule. So you'll tend to have loop regions at the surface, that's true, and they will tend to be highly variable. So if you make an alignment of a lot of proteins, you'll find some bits that don't align very well, that have they can be bigger or smaller, or their amino acids are just different every time. You're probably looking at loop regions when you see those, yeah. And so you can start to infer what it is you're looking at. There are patterns in loop regions. So loop regions do fit onto the Ramachandran plot, and there are a limited number of psi and phi angles that can actually be allowed. Loops allow the polypeptide chain to change direction. And the real common one is a beta turn, which is a turn where we're between two elements within a beta sheet. Those occur quite a lot. So, random coil is everything else, but potentially including beta turns. Beta turns, you can say, I would say they're part of random coil, or you can mark them out separately. Most prediction software does not predict beta turns. You have to look at those by eye and recognize them. But it'll use the concept of a beta turn in predicting where the beta sheet ends, or the beta strand ends. So. And then there are flexible loops, which are just bits that are going to be fundamentally floppy, that can fold up into many different shapes, and probably do sort of change shape willy-nilly as the protein exists, as it's floating around inside of a cell. Flexible loops can sometimes become rigid when they bind to and hydrogen bond with the right protein. So they can be like a key that won't unlock and won't adopt a particular conformation until the right molecule comes along. Yeah. So that's the idea behind your flexible loops. All right. So yeah. All right. You got this. I'll leave the quiz up there. You can have a look, see how you do. It's a good thing we can talk about in class. And yeah, I think that's all we need. You can find the answer to number 4 in the video on alpha helices. So have a look at that. Yeah. Yeah. I think we're good. Thanks.